Do athletes really need to eat a high carbohydrate diet? Or can athletes eat a low carbohydrate, high fat diet to train their bodies to run on fat as their primary fuel? In today's lesson, we take a look.
Ketogenic diet has neurological benefits. Why do we have to eat such an enormous amount of food?
Complex science, clear explanations. Class is starting now.
Hi, I'm Dr. Chris Masterjohn of chrismasterjohnphd.com, and you're watching Masterclass with Masterjohn. We are now in our 17th in a series of lessons on the system of energy metabolism. This week, we're talking about the relative ability of carbs versus fats to support athletic performance, and we're splitting this into two lessons. In today's lesson, we talk about the principles involved, and in the next lesson, we'll talk about the evidence. So let's dig right into the principles. As shown on the screen, in exercising skeletal muscle, there are three primary systems of energy metabolism. All of these have as their goal supporting ATP production, because ATP is what is directly utilized to contract your muscles. However, in the phosphagen system on the left, we see ATP listed. And that's because when you contract your muscle, there's an existing pool of ATP that can be used immediately. That immediate ATP that doesn't need to be formed through another process is the ATP that's listed as part of the phosphagen system. The other component is creatine phosphate. Creatine phosphate, like adenosine triphosphate, is a donor of phosphate or phosphoryl groups. In the case of ADP, the phosphoryl groups are used to directly stimulate muscular contraction. In the case of creatine phosphate, as we'll see in the next slide, the phosphoryl groups are used to recycle ATP. Nevertheless, because ATP and creatine phosphate are both direct donors of phosphoryl groups, in other words, they are the genesis of those phosphoryl groups, we call this the phosphagen system. The second system is anaerobic glycolysis, which we've already talked about in detail, where glucose is converted to lactate, generating a small amount of ATP. And anaerobic glycolysis and the phosphagen system together make up the anaerobic component of the energy supply. Some sources will divide the anaerobic component into a lactic subcomponent and an alactic subcomponent. The lactic component of the anaerobic supply refers to anaerobic glycolysis, and the alactic component of the anaerobic supply refers to the phosphagen system. Those simply refer to the fact that we don't make lactate in the phosphagen system, and we do in anaerobic glycolysis. The third system is oxidative phosphorylation. As its name implies, we use oxygen to phosphorylate ADP to make ATP. That happens in the electron transport chain, where oxygen is the final electron acceptor of the electrons from the food that we ate. It derives the energy in those electrons to make ATP, and those electrons were harvested in the citric acid cycle, ultimately taken from fat, carbohydrate, or protein. That's the aerobic supply, and it requires oxygen. Everything on the left is the anaerobic supply. It can operate in the absence of oxygen. Notice that all three macronutrients support the aerobic supply. But only glucose is supporting anaerobic glycolysis, giving carbohydrate a special role in supporting anaerobic energy metabolism. The role of creatine is as a temporal and spatial buffer of the ATP pool. Temporal means across time, spatial means across space. The enzyme creatine kinase catalyzes a reversible reaction where phosphocreatine can phosphorylate ADP to make ATP, itself becoming creatine, or creatine can take the phosphoryl group from ATP, becoming phosphocreatine, generating ADP. This reaction happens both ways and both ways are equally important to the use of creatine as a buffer of the ATP supply. However, the equilibrium strongly favors ATP production. And what that means on a practical level is that as ATP transiently declines, you will preferentially use phosphocreatine to regenerate the ATP pool. So if you have intense exercise and you see these metabolites declining, you'll see phosphocreatine decline far earlier than you'd ever see ATP decline, because phosphocreatine gets used up to maintain the ATP pool as normal. Nevertheless, you will reach a point where, as the phosphocreatine concentration declines, as a reactant in this reaction, that slows down the phosphorylation of ATP. And you will reach a point where the phosphorylation of ADP to make ATP versus the phosphorylation of creatine to make phosphocreatine is happening at equal rates. So phosphocreatine isn't useful until it gets to zero. It's only useful until that equilibrium value is achieved. Because at that point, you're not creating ATP any faster than you're using it. We'll see in later slides that phosphocreatine seems to be useful right until the point where it's depleted to 40% of the original value. Now, as shown in the upper right, there is 3 or 4 times as much phosphocreatine as there is ATP. So we can say that phosphocreatine supplies about 75% of the phosphagen pool. The fact that it's so much more abundant allows it to be more broadly and more densely distributed in the cell. And that means that when ATP is used up, there's almost always going to be phosphocreatine nearby that it can have immediate access to get regenerated, reducing the amount of time that it would take to get regenerated in the mitochondrion and the amount of space that it would have to travel across to make that recycling happen. We see this played out in space in the bottom panel of this diagram. As ATP is utilized for muscular contraction, it generates ADP. Phosphocreatine is right there to recycle it. It doesn't need to travel all the way back to the mitochondrion or even necessarily get ATP from glucose, or get phosphorylated in glycolysis, I should say. Now that phosphocreatine is given up a phosphoryl group, it needs to be recycled. It has the quickest access to glycolysis, which is immediately there. But remember, glycolysis supplies a much more limited amount of ATP than can be produced in the mitochondrion. So some of that creatine can be quickly regenerated with glycolysis, or some of it may have to travel back to the mitochondrion where it can be regenerated. When it gets regenerated, it's using up ATP. So what's happening is you're buying time and reducing the amount of space that you need to travel rather than any net difference in ATP. In other words, you're using ATP to support phosphocreatine that's then recycling ATP. All you did was translate energy from one ATP to another using phosphocreatine as an intermediate. There's one really important point that needs to be drilled home here, and that is that many sources misleadingly present the 3 systems of energy metabolism as being sequentially used during exercise, as if you use phosphocreatine, then you use glycolysis, then you use the aerobic system. You are always using phosphocreatine, always. I'm using phosphocreatine right now to get the energy that I need to move my hands in the air as I communicate to you with my hands. That's using up phosphocreatine. Even at rest, you're using phosphocreatine. And in fact, you're using phosphocreatine that's being supported by ATP from glycolysis or by ATP from oxidative phosphorylation. We'll clarify this momentarily, but when we say that phosphocreatine is important in early exercise, what we mean is that its net contribution to how intensely you can exercise is what's important there. We don't mean that it's used up first before we start using the other systems. If we look at the spatial arrangement of these different supplies of energy, we can see that muscle has access to many different types of energy, but some of them are much closer to the spot where it's needed than others. So let's say over here is where we're using up ATP. Phosphocreatine is right there at the site of the action. Glucose being converted to pyruvate in the cytosol is not far away, and that can generate ATP to recycle the creatine or to put into the direct supply, but its contribution is relatively limited. Then we have mitochondrial metabolism. Whether it's coming from fat or carbohydrate, we have to transport energy molecules into the mitochondria, make the ATP, and transport the ATP back out into the ATP pool. That takes time. That takes resources for transport. But when we do that with pyruvate, we're mostly getting it from glucose that came from glycogen. The glycogen is in the cytosol of the muscle cell. Yes, it has to be broken down, and that takes time, but it's very close to the site of the action. We can contrast this with fatty acids, which are primarily coming in the blood from adipose tissue. We can even contrast it with the minority of glucose that's coming from the blood that came from the release by the liver. Both of these have to be released by other organs, be released into the blood, be transported to the muscle, be taken up into the muscle, be transported into the mitochondrion before they can make the ATP that then needs to come out and equilibrate into the pool of ATP being used for muscular contraction. That takes a lot of time. Now, not shown here are intramuscular triglycerides that are closer to the action. We'll talk about them briefly later, but less is known about them, and the evidence we have indicates that they're not a major source of energy during high-intensity muscular contraction. Shown on the screen are quantitative estimates of the speed with which you can use these sources and the amount you have available that are adapted from the source listed at the bottom. In the middle column, we're looking at the maximal rate of ATP hydrolysis measured in moles per minute. Moles is a unit of number. That means how many phosphoryl groups can you hydrolyze per minute. And this doesn't mean 4 phosphoryl groups, this means 4.4 moles per minute. If you want exactly what a mole means, you can Google it, but it's a unit of the amount per unit time here. You can see as you go down the list here that this number is highest for the phosphagen system, reflecting the fact that it's closest to the action and is used most rapidly. As you go down, you see that the number gets lower and lower, meaning the supply is slower. So muscle glycogen to lactate, to speak of ATP hydrolysis in moles per minute, means you make glycogen into the lactate, you generate ATP, then that ATP is available to hydrolyze. How many phosphoryl groups could you hydrolyze from the ATP derived from that process? How fast could you do all of that? And that is 2.35, so it's about half of the rapidity, the speed that you get from the phosphagen system. If you're turning the glycogen to carbon dioxide, you're transporting stuff into the mitochondria, doing a longer process, taking the ATP out, that takes longer. You have 0.85 to 1.14. Let's say it's about 1 or 1.14. We can simplify this. Let's say 4 to 1 to round it off. Well, each time we go down to greater distance from the site of the action, we're cutting the speed of the supply in half. That's a huge difference. The reason that they give a range 0.85 to 1.14 is because it's thought that the glycogen being in the muscle cell, the limiting factor for the speed with which you use it is the degree to which you can get oxygen into that tissue. So well-trained individuals who use oxygen really efficiently can do that faster. By contrast, if you look at liver glycogen to carbon dioxide or fatty acids to carbon dioxide, you're cutting this rate to 1/10 of what you get from the speed of the phosphagen system. They don't give a rate because it's thought that in that context, it's not your training and oxygen diffusion that's the limiting factor. It's actually the hormones and other factors that are going to govern the release of the rate of release of glycogen from the liver, the rate of release of fatty acids from adipose tissue, and their transport to the muscle. Given that, it's also interesting that there's very little difference between carbohydrate coming from the liver and fatty acids coming from adipose tissue. That again reflects the fact that the rate, the speed, is not so much about carbs versus fat. It's more about the travel time from one tissue to another. Now, if you look on the right column, you see the amount of ATP supported in moles. Here, there's primarily an inverse relationship between speed and abundance. So the phosphagen system is the fastest, but there's the least amount available. As you go from muscle glycogen to lactate, you get a little bit slower, but there's 10 times more of that available. As you go muscle glycogen to carbon dioxide, it gets even slower, but the amount is over 10 times more. Now, liver glycogen is an exception to this relationship because your muscles just store a lot more glycogen than your liver does. So there's not as much available and it's slow. But if you look at fat, fat is slow, and yet there's this enormous quantity of fat. Even a lean individual has 30 times more fat than they have glycogen. So we're talking about an enormous amount of energy that's very slowly burned when we're talking about deriving fatty acids from adipose tissue. On the one hand, that makes you think it would probably be good for endurance to run on fatty acids. On the other hand, it would probably be really bad for peak intensity to run primarily on fatty acids simply because they cannot produce the speed. If the phosphagen system and anaerobic glycolysis are both providing speed, then it makes sense that we want to think more about what causes us to rely on anaerobic glycolysis instead of phosphocreatine, because it's phosphocreatine that really has this speed. And it's phosphocreatine that isn't going to require carbohydrate. So let's take a look at the principles of phosphocreatine depletion, because when it gets depleted, that's when we're super reliant on anaerobic glycolysis. Now, the data on the screen are looking at phosphocreatine depletion during knee extension exercises where they were done in a way that would lead to exhaustion within 3 minutes. In this paper, they were investigating the concept of critical power. The idea of critical power is there's some level of power output that you can maintain almost indefinitely until your energy system just totally tanks. But there's some limited ability for you to work at a level that's way higher than that. That level that allows you to go on and on and on like the Energizer Bunny, that's the critical power. And what they're doing here is saying, okay, what happens when you exercise way above your critical power to the point where you get exhausted? When you get exhausted, that doesn't mean that you couldn't do any more work. It means you can't do any more work at that rate well beyond your critical power that you're asking to do. So these people couldn't do the knee extensions at that rate anymore, but they probably could have gotten up and walked over to the refrigerator or something like that. And what you see is that the phosphocreatine supply declines to about 40% at the point of exhaustion. Now, does that mean that anaerobic glycolysis isn't supporting your work above critical power that got you to that exhaustion point? Does it mean that oxidative phosphorylation isn't operating to help you reach that peak power that got you to the exhaustion point? No, because anaerobic glycolysis and oxidative phosphorylation are both supporting the recycling of creatine to phosphocreatine. So this doesn't mean that you just used phosphocreatine until you got to this point. It means that you were using phosphocreatine up faster than you could recycle it. And therefore, its concentration declined despite the fact that you're supporting recycling with anaerobic glycolysis and oxidative phosphorylation. And when it declines to about 40%, it reaches a point where it's being recycled by ATP just as fast as ATP is recycling it, and there's no net benefit of the phosphocreatine to support that extra work. And as you lose this immediate buffer to the ATP pool, you lose the ability to operate at that peak of power. They then took these subjects and they had them recover at 3 different workloads. One was 80% of critical power. They were able to go on for 1 minute and then they couldn't do it anymore. And so the triangle stopped. They also went at 50% of critical power, or at 0%, meaning complete rest. You can see that at this level where it took 3 minutes to exhaust the phosphocreatine supply, at rest, it took 3 to 4 minutes to get the bulk of the recovery of the phosphocreatine pool. But even after 10 minutes, you kept increasing your recycling of the phosphocreatine pool and you still weren't 100% there. But from a practical level, you see that you're really getting the hugest bang for the buck after 3 or 4 minutes of rest. If you are operating at 50% critical power, you recover along the same time scale, but you never reach the same degree of recovery of your phosphocreatine pool. So you really need complete rest or something close to it for 3 to 4 minutes to get the bulk of your phosphocreatine regeneration. Now, if we're looking at athletic performance, usually we're not looking at total, consistent, exhaustive work over 3 minutes. Usually we're looking at bouncing back and forth between work, rest, or something in between. And in the data that are shown on this screen, we're seeing interval work where knee extensions were done at a power that was estimated to produce exhaustion after 6 1-minute intervals alternating with 30 seconds of rest in between each interval. And you see that in each 1-minute interval, you have a big decline in phosphocreatine. During each 30-second recovery period, it goes up. During each work period, it goes down again. But you never have enough recovery in 30 seconds of rest to get up. So what happens after 6 cycles to produce exhaustion is that your creatine goes down, only partially recovers, goes down even further, only partially recovers, goes down even further, and then by the 3rd cycle, you're really nearing that 40% level that's going to produce exhaustion. And so somewhere around this 3rd, 4th, 5th, or 6th cycle of work, you're going to hit that point where you're just not recovering enough creatine to go very hard after that little bit of rest. As I had said before, though, it's not you use phosphocreatine and then you exhaust it and then you use the other energy systems. Here's data looking simultaneously at the use of the energy systems over 24 seconds of maximal voluntary contraction in knee extension exercises. And what you see is that in the first few seconds, you're getting all this energy from phosphocreatine, and then its contribution declines. You are using oxidative phosphorylation from the very beginning. And you're using anaerobic glycolysis after a lag of several seconds where it then starts increasing. At the 15-second mark, those 3 energy systems converge to where they're all equally important. But even here, where you're using up phosphocreatine, to the degree that oxidative phosphorylation and glycolysis come in, they are helping your creatine supply recover and preventing it from falling as fast as it otherwise would. They're both operating at the same time, so you're going to get maximal phosphocreatine regeneration. If you have both of those operating, it's not one versus the other. The more ATP produced by those systems, the longer phosphocreatine is going to take to get depleted, and the longer time it's going to take to get the 40% depletion that it takes to lose your ability to go at that really high peak power. The data on the screen strongly suggest that there's always going to be a time contribution where in a short period of time, you are going to require the anaerobic systems. What's really fascinating about this data is it represents over 30 studies, where the result of each study is an individual system that's looking at the percent contribution of the aerobic system over time measured in seconds. And what's particularly fascinating is that some of these studies only lasted 15 seconds. Other studies lasted over 1,600 seconds, going well beyond the point that's showed on this graph. And despite the fact that there was such difference in the time under which exhaustive exercise was studied, and therefore the intensity, what's remarkable about that is that there's a general fit across all these studies to this curve that implies a relatively tight estimate of the amount of time it takes to go all anaerobic. And that suggests that no matter what your intensity level is, there's always a time component to going aerobic. And that shouldn't be that surprising, because to get that exercise relying on aerobic metabolism is not just about how well you trained and what you ate. It's about the unchangeable fact that you need to start breathing faster to take in more oxygen, and your heart needs to start pumping faster to get the oxygen from your lungs to your muscles, and your muscles need to take it up. And that's a lot of processes that need to happen to adjust to the new rate of work that you're doing to become aerobic. Another thing that's quite stunning about this is that you never truly reach 100% aerobic during this time frame. So that implies that even beyond 500 seconds, which is almost 9 minutes, you are always using a substantial amount of anaerobic metabolism. And your phosphocreatine supply is very likely burned through, and that gap is going to require reliance on anaerobic glycolysis. The data on this screen were taken from endurance-trained or sprint-trained athletes who did 90 seconds of all-out cycling. And the key takeaway point from this graph is that when we looked at aerobic proportion increasing over time, that corresponds to the decrease of intensity that you get by the time you've gotten to that point. So the key line to look at in this graph is that energy demand, meaning how hard are you working to need ATP support, peaks at the very beginning and then declines as you get more exhausted. This is the exhaustion of the power output above that critical power threshold, and then you decline to that critical power, that level that you can sustain over a long period of time. And if you look at the dotted line for endurance trained or the solid line for sprint trained, you'll see that the sprint trained, unsurprisingly, had higher peak energy demand because they're doing higher rate of peak work. And if you look through this data, you will see that in general, sprint-trained athletes were better able to tap into glycolysis and endurance-trained athletes had lower peak intensity and they were better able to peak into aerobic metabolism. But the point I want to make here is that by the time you're getting mostly aerobic, the phosphagen system has well declined. You've moved into the area where you're relying to a very substantial degree on glycolysis. And by the time you adjust to aerobic metabolism as a primary support of the energy supply, you've moved well beyond the point of the peak power. That means that this power peak is always very reliant on anaerobic glycolysis. If you look at what it takes to transport fatty acids into muscle cells to burn for energy, it requires more vulnerabilities than burning carbohydrate. So look, for example, at what has to happen. Albumin is a protein in the blood that has to take fatty acids that are hydrolyzed from triglycerides in adipose tissue. Albumin carries the fatty acids to the cell where fatty acid binding proteins, in a series of events that are glossed over here in their details, take the fatty acids and transport them into the cell. Coenzyme A needs to make a fatty acyl-CoA to use the carnitine shuttles to get inside the mitochondrion. We'll go into this in much more depth later when we talk about fatty acid metabolism. The fatty acyl-CoA then delivers the fatty acyl group to beta oxidation, where coenzyme A is used again to come in to get each acetyl group that's burned in cellular respiration. If we're burning glycogen, first of all, our glycogen is in the muscle cell, so we don't need to transport through the blood. Second of all, glycogen releases glucose that goes through glycolysis to become pyruvate, and pyruvate is carried through the mitochondrial pyruvate carrier to get into the mitochondrion. Pyruvate does not require coenzyme A to get into the mitochondrion. Furthermore, if there's any limitation in the mitochondrial pyruvate carrier, pyruvate gets converted to alanine, and alanine gets transported into the mitochondrion through a mechanism that we know exists but we don't know the details of, and gets converted back into pyruvate. So transporting pyruvate into the mitochondrion could become limited, but there's a backup mechanism to get it in there. Pyruvate then undergoes decarboxylation to acetyl-CoA, where coenzyme A is used for the first time, and the acetyl-CoA enters cellular respiration. We can think through a lot of potential limiting factors here. For example, maybe something is limited at adipose triglyceride lipolysis. Maybe the carrying capacity of albumin is limited. Maybe the capacity of fatty acid binding proteins are limited. Maybe the CoA pool is limited for either of these things, but for fatty acids we have to consider maybe the cytosolic CoA pool is limited, and that is going to hurt entry of fatty acids into the mitochondrion. If there's any overwhelming of this transport system, there's no backup like there is for pyruvate. Furthermore, although not directly shown in this diagram, this pyruvate is also the source of anaplerosis in the muscle cell. So if oxaloacetate becomes limiting to burn acetyl groups, you can get that from pyruvate, and you can't get it to anywhere near that degree from fatty acids, as we discussed in the last lesson. So we can get two takeaway points from this. First of all, there are a lot of things to consider that could be fat adapted or carb adapted depending on your chronic diet. For example, maybe fatty acids upregulate lipolysis at adipose tissue, upregulate albumin's carrying capacity, upregulate fatty acid binding proteins, upregulate the carnitine shuttle, etc., etc., etc. Many things that could be fat adapted here. On the other hand, if you're going to burn more fat and less carb, you can easily hypothesize that there would be more transport vulnerabilities to the fat that would limit its ability relative to carbohydrate to keep energy metabolism going. But finally, any of these things can be overwhelmed. the mitochondrial pyruvate carrier could be overwhelmed, and the alanine transport could be overwhelmed, and then it would be beneficial to have fatty acids. So you're always going to get maximal power if you maximally use all of these potential fuels simultaneously, because it allows a more robust ability to get around any possible limitations in transport or utilization. There are some other sources of fatty acids for the muscle that we've glossed over at this point, and that's because the available evidence indicates that they're not very important, particularly for high-intensity exercise. So VLDL triglycerides have a small ability to get metabolized by lipoprotein lipase, or LPL, in the muscle cell to release their fatty acids. But they have a very low affinity for muscle LPL and don't seem to be a very significant source of fatty acids to muscle cell. Chylomicrons have 50-fold higher affinity for muscle LPL than VLDL triglycerides. Although they have high affinity, they're at much lower concentrations in the blood, and in fact, they only transiently appear after meals. In order to utilize chylomicron triglycerides as a source of fatty acids for exercising skeletal muscle, you'd have to eat a high-fat meal right before you exercised. That hasn't been studied well, but most people who are athletes would probably agree that eating a really big high-fat meal is not necessarily what you want to do right before you exercise. Finally, there are intramuscular triglycerides. They do get used during exercise, but they actually get used at a lower rate in high-intensity exercise than low-intensity exercise. So they don't seem to be a useful support for fatty acids to high-intensity exercising skeletal muscle, despite their close proximity. And some people have speculated that the main role of intramuscular triglycerides may be to sequester fatty acids as an interplay so that fatty acids don't accumulate and have toxic effects beyond their rate of utilization. Regardless, they're not utilized well in high-intensity exercise. And in fact, in McArdle's disease, which is a genetic disorder of your ability to tap into your glycogen metabolism, even then, you have this severe wall that you hit in early high-intensity exercise as a result of the genetic disorder and the ability to access glycogen. And even during that time, even they don't tap into their intramuscular triglycerides in a way that compensates. So these are all very minor sources of fatty acids. Finally, I should note that I didn't even show ketones on this diagram. On a ketogenic diet, you do transfer your energy metabolism towards ketones. Ketones are small, water-soluble metabolites of fatty acids, but they're preferentially utilized by cells that aren't good at using fatty acids, such as, for example, the brain. Ketogenic, even advocates of ketogenic diets for exercise, such as Jeff Volek, whose recent paper we'll look at in the lesson next Thursday, acknowledged that muscle cells during exercise are mainly using fatty acids rather than ketones, even on a ketogenic diet. So finally, to synthesize all this information into its practical conceptual implications, we can imagine two requirements for anaerobic glycolysis, and I'm referring to those as the temporal lactic requirement and the intensity lactic requirement. These aren't technical words used in the literature. I'm just using these phrases to myself to communicate these concepts. This scale is not to scale. There's no units in the time axis. This is just saying that over time, you're going to relatively quickly deplete your phosphagen supply. How fast that happens depends on how intense your exercise is. It's going to take time to get maximally aerobic. Surprisingly, that depends a lot less on how intense your exercise is, because that's mostly about the facts that you need to adjust your breathing rate, your heart rate, and your supply of oxygen to muscle in order to get aerobic. At some point in very early peak intensity, you don't require very much anaerobic glycolysis, especially in the first 5 seconds. It hasn't even really kicked in to be able to support anything. And so in very quick exercise of very high intensity, you do not have a lactic requirement, and that's indicated by this sliver where you're using completely phosphagens to supply that intensity. But at some point you run out of the phosphagen pool and you take time to get to the ability to rely on aerobic metabolism. The gap between those two events is the temporal lactic requirement. Again, my words. And even after you reach 100% of your aerobic capacity, you're always going to be able to push it higher using anaerobic metabolism. But if you're at the point where you've depleted your phosphagen supply, that increase in intensity has to come from anaerobic glycolysis. Therefore, carbohydrate is always going to make a specific contribution to intensity, one that's associated with a time gap between the phosphagen depletion and the aerobic adaptation, and one that's associated with the peak intensity that you can always get when you go beyond what your aerobic metabolism can deal with. Fat adaptation or keto adaptation, by physiological principle, cannot change those lactic requirements. Fat adaptation can change the relative proportion of carbs versus fat that you're using to supply aerobic metabolism. So you can fat adapt this window here. But when you do that, you do it by consuming less carbohydrate. So if you get better at burning fat at the expense of using carbohydrate, is that net benefit? Theoretically, it could be when your requirement is to go long instead of to go intense. Theoretically, there could be some balance where you can go longer at a relatively high intensity when you're burning fat, but not to your highest intensity when you're burning fat. But even that is just a hypothesis in need of testing, because that fat adaptation will come at the expense of carbohydrate metabolism. And you clearly get the most energy for the longest when you're able to use all of the fuels simultaneously. So what does the evidence say about that ability for fat adaptation? We'll look at that in the next lesson. The audio of this lesson was generously enhanced and post-processed by Bob Devodian of Torian Mixing, giving you strong sound and dependable quality. You can find more of his work at torianonlinemixing.com. You can continue watching these lessons on my YouTube channel at youtube.com/chrismasterjohn, or on my Facebook page at facebook.com/chrismasterjohn, or you can sign up for MWM Pro to get early access to content, enhanced keyword searching, self-pacing tools, downloadable audio and transcripts, and hyperlinked further reading suggestions, putting a whole world of information right at your fingertips. Plus, there's a community with a forum for each lesson. So if you really want to own the lessons and get the most of them and really study them, all those tools are at your disposal if you sign up for MWM Pro at chrismasterjohnphd.com/pro. All right, hope you found this useful. Signing off, this is Chris Masterjohn of chrismasterjohnphd.com. You've been watching Masterclass with Master John, and I will see you in the next lesson.